Okay, well, thank you for those definitions. So let's talk about individual ones first. We're going to talk about Illumacell first. Can we spend about 5 minutes on Illumacell?
Yes, sure. Yeah.
Okay, let's do that.
Okay, so Illumacell is a company that's, I have a couple different companies, and Illumacell is, I founded that company, is directly involved in trying to advance using light to activate a drug and kill cancer cells. Now, the drug has actually been out. There's about 25 different photosensitive drugs out there. But the one we really like is one that has the longest track record and is actually, in a sense, the weakest of the drugs, but it works really nicely. So what happens is you take this drug orally, and sorry, I should back up. So right now they're doing light treatment with low power red light of the right wavelength or blue, and you put topical cream on your skin, and then you shine the light on it, and wherever the cancer cells are, they will be killed off, and you end up with a perfect cosmetic look to it, because instead of burning the area with liquid nitrogen or using some of the chemo type creams, you're not burning the whole area. You're selectively killing the cancer cells, and it works really nicely. I mentioned to you earlier that the compound that's created in your body. So you take this drug or topically, and then 4 hours later, it converts to porphyrins. Porphyrins are a certain type of molecule that, interesting enough, that molecule, the next step in your body, everybody's got this compound in their body called 5-ALA and delta aminoalluric acid. But anyways, what happens is the delta aminoalluric acid is the precursor to creating porphyrins. And then the porphyrins, the next step in your body is the iron molecule goes into it and makes it a stable molecule and goes on to provide the iron in your blood or the heme in the heme. So that's how that process works. Now, what happens is, interesting enough, is if you take an excess of this, so orally or topically on the area, the cancer cells create a lot more porphyrins than the normal cells. And then when you shine red light on it, or different wavelengths, but red is one of the best lights, there's a specific wavelength, it will cause that molecule to break apart. And as before it becomes the iron in your blood, just before that breaks apart and releases a singlet oxygen molecule right inside the cancer cell. So it's like pouring almost hydrogen peroxide in each cancer cell. And it does a really nice job of selectively killing the cancer cells, as opposed to the normal cells around there, which can take a certain amount of oxidative stress, and they can easily deal with that. And so you get a very selective occurrence. And what we've done differently is, there was a picture up earlier, and I'll just show you the real thing. This here light head is something I've got patents on. There's 1,000 high power LEDs on here, on that light head on each one. And 3 of those, just to give you an idea, 3 of those LEDs would give you the equivalent of a 100-watt light bulb. And we have 1,000 on one light head and another 1,000 on another. So basically almost like 500 100-watt light bulbs are going off at the same time. And so we literally can go halfway into the body without burning the skin, non-invasively, going in and breaking apart that molecule inside the cancer cells, so we can deal with solid tumors. Not so much on the blood side. We haven't gone there, but we have now a lot of time on that side of things with tumors and that. On an anecdotal, plus we've gotten certain governmental approvals to do single off trials. So that's the cancer side.
That's super cool. So the pharmacist in me wants to know what drug you take. I'm putting you on the spot. Can you tell me what drug that is?
Yeah, it's 5. So there's quite a number of them, but some of them got a really bad rap because they've made the porphyrin directly, and then you can't be in the sun for 2 months, or you'll have second or third degrees burns. So they went to that drug because they had so little light. They needed to increase their drug strengths or the power of the drug. We've done just absolutely the opposite. We use this very low energy, low side of the drug. There we go, the low side of the drug, and a very high power light, and we get the results that we want. And the neat thing is, after multiple treatments, you're creating an immune response, so the immune system learns the cancer, and good things happen. But it's not a one-shot treatment unless it's something on the level of the skin. And that's been out there for now over 25 years. The drug taken orally is kind of unique, although it's been used for glioblastomas, for detection of, so when the kids, they did this in England. There's over 500 kids that took the 5-ALA 4 hours ahead of time in the drink. And then when they opened up the skull for the glioblastoma, they could shine, after they debulked most of the tumor, they could shine one wavelength of light down, a UV colored light, and whatever fluoresced red, it would illuminate red, and they'd know that's still cancer remaining. That's how accurate this is, and they would go then and use it more. So they get better outcomes on glioblastomas. So the track record of this, because it's a natural stuff within your body, is fantastic, because it's something your body can't get used to. The cancer can't get used to it, and it comes at different angles, and it's totally destructive. So that's the cancer stuff. I love it. I wish we could see this thing just take right off. Unfortunately, there's a lot of people that would still like to keep the money stream going on the other front. And all we would need is somebody with a large, deep pockets to be able to help us to run a study, which, $1 million to a drug company is nothing, 5 million, 10 million. But right, to a device company, that's a lot of money. So we need $1 million to run a trial, is basically what we need. And there's not a lot of people that want to go and invest in $1 million on something that there isn't any, the research isn't big on yet. So